Hello, my project is can zinc nanoparticles and silver nanoparticles serve as antimicrobial agents against E. coli K12? Antibiotics are the chemically synthesized or drugs that we use to kill pathogenic bacteria that causes infectious diseases. We can see them do this in the picture to the left, where the white outlines represent discs containing the antibiotic, and the black spots represent the areas where bacteria is no longer growing. This is called a zone of inhibition. However, over the past few years, these pathogens have developed mechanisms to fight the antibiotics that we use to kill them. It causes about 23,000 deaths a year and billions of dollars just in the US. Unfortunately, there is not a lot of innovation because it is not a very profitable business. Due to the many mechanisms they have to prevent the antibiotic from inhibiting their growth, new ones are arriving, and they're usually naturally derived due to the lower toxicity and higher stability of these. Nanoparticles have been looked at as possible novel antibiotics due to their ability to be engineered in size, surface layer properties, and ability to be optimally specific in the delivery of active agents. Biowaste has also been thought of as a possible source of nanoparticles due to previous research showing their green synthesis as being fast, eco-friendly, inexpensive, and non-toxic, not to mention that they are a sustainable, renewable energy source that can be easily obtained. Zinc nanoparticles specifically are beneficial because of the amount that is readily available. Photochemical activity has been observed in it, there's semiconductors, and previous research has shown antibiotic activity. Silver nanoparticles have similar reasons to being looked at as well. So therefore my purpose is to use biowaste to determine if biowaste-derived activated zinc and silver nanoparticles can inhibit the growth of gram-negative E. coli K12. These are my For the nanoparticles, I will first take samples of each of the peels, use a mechanical grinder and sieve to create minute particles, and a SOXA extractor and rotary evaporator to create minute particles and a final reducing agent. Then zinc nanoparticles and silver nanoparticles will be synthesized through the filtration of aqueous silver nitrate and zinc nitrate using sodium hydroxide and a centrifuge. To grow the E. coli, I will simply transfer liquid culture to broth using a micropipette, allow it to grow overnight in an incubator, then transfer it to an agar plate using a cell spreader. For the diffusion assay, I will sterilize the paper discs, and then I will saturate them with each of the biowaste sources. Then I will place them on the E. coli K12 plates, allow them to grow, and I will do this for the control as well for 3 trials. This is how I will record my data. It's basically just showing the percentage of activity and the length of the diameter of each of the discs for the zone of inhibition. This is my second set of data tables, and this is third, and this is for my control. For the analysis, I hope to just see which one has the highest percentage compared to the control, compared to the other biowaste sources, and whether zinc or the silver nanoparticles had higher percentages than each other, or if they were similar, things like that. For the conclusion, I hope to basically just find any zone of inhibition that can work as a potential candidate, because then I can optimize the procedures to make it more efficient as an antimicrobial source. Future research and my references.